So, welcome to today's session of plant physiology and metabolism course. We have gone into details of uh, glycolysis pathway, its regulation, and then how pyruvate is converted into acetyl coenzyme A. So, before going into details of tricarboxylic acid cycle or Krebs cycle, it is basically very crucial that would to discuss these aspects in understanding that how basically tricarboxylic acid cycle functions, and then we will explore into the details of TCA cycle and comprehend each step in detail. Now basically tricarboxylic acid cycle, also known as the citric acid cycle, just or Krebs cycle, named after its discoverer Hans Krebs, basically consists of 8 successive reactions, and these reactions include formation of citrate, formation of isocitrate through cis aconitase, then oxidation of isocitrate to alpha ketoglutarate, and then release of carbon dioxide. The oxidation of alpha ketoglutarate to glutarate to succinyl coenzyme A, and release of carbon dioxide, conversion of succinyl coenzyme A to succinate to fumarate, and then hydration of fumarate to malate, and finally the oxidation of malate to oxaloacetate. So, this is the different steps of TCA cycle in nutshell. Then we will go into further details of these steps, how these steps are catalyzed by specific enzymes. So, this is very interesting to share with you. Now, the first step, that is the first reaction of the TCA cycle, is the condensation of acetyl coenzyme A with oxaloacetate to form citrate, and this step is catalyzed by citrate synthase. You see in this reaction in the slide. So, this is the first reaction of the TCA cycle. Basically, this reaction involves the attachment of the methyl carbon of the acetyl group to the carbonyl group at position at carbon 2 of the oxaloacetate. Then citroyl coenzyme A is basically the intermediate that is created within the active site of the enzyme. So, basically due to the overall delta G of this particular reaction makes this step particularly irreversible under standard condition. Then citrate synthase is basically present in mitochondria and glyoxysomes in plants, playing a very, you see that a crucial role in the glyoxylate cycle. The mitochondrial enzyme is basically homodimeric with 2 domains in each subunit, one large and rigid, another small and flexible. So, you should note that the mitochondrial citrate synthase is basically homodimeric, and you must also remember that of these 2 units, one is large and rigid, another one is small and flexible. The active site of the enzyme lies between the 2 domains, and conformational changes occur upon substrate binding, leading to the formation of citroyl coenzyme A. Then citrate synthase is basically regulated and inhibited by ATP, although generally the inhibition is not very significant due to low ATP concentration prevailing in the mitochondria. Then NADH, which is a product of TCA cycle, and succinyl coenzyme A are basically serve as allosteric inhibitors of enzyme citrate synthase. You must remember that these NADH and succinyl coenzyme A, basically these two intermediate serve as allosteric inhibitor of enzyme citrate synthase. This is very interesting to share. Now, second step of the TCA cycle, that is catalyzed by aconitase. You see in the reaction, citrate is converted into isocitrate. So you see that conversion of citrate into isocitrate basically catalyzed by enzyme aconitase. So basically this is converted into a two-step with sort of having a intermediate. So the reaction involves the conversion of symmetrical tertiary alcohol, that is citrate, to an asymmetric secondary alcohol, isocitrate. The activity of enzyme aconitase, basically the active site of enzyme aconitase basically contains an iron sulfur cluster, and cysteine residues form generally coordinate bonds with 3 iron atoms, and water is removed from citrate to produce cis aconitase, which is then rehydrated to form isocitrate. Generally, aconitase is present in both mitochondria and cytosol, participating in TCA cycle and glyoxylate cycle. In the cytosol, you see that aconitase along with NADPH dependent isocitrate dehydrogenase provides 2-oxoglutarate for amino acid biosynthesis. This is also very crucial. You must note it here. Then aconitase catalyzes the first step in the citric acid metabolism during the ripening of the fruit. You see that one compound, fluoroacetate, is a poisonous agent that blocks the tricarboxylic acid cycle in vivo condition by affecting this enzyme aconitase. So basically fluoroacetate serves as an inhibitor of enzyme aconitase. Now next step is, you see that oxidation of isocitrate to alpha ketoglutarate. Basically isocitrate undergoes oxidative decarboxylation, resulting in the formation of alpha ketoglutarate, and this reaction is accompanied by reduction of NAD to NADH and the release of one carbon as carbon dioxide. And NAD isocitrate dehydrogenase basically catalyzes this two-step reaction, which involves the synthesis of an intermediate compound called oxalosuccinate. You see that in the slide, in the first step, what happens, the hydroxyl group, that is shown with colored mark, hydroxyl group located at second carbon. You see in the slide, extreme left, the hydroxyl group located at carbon number 2 of isocitrate is basically oxidized to generate oxalosuccinate. This is basically followed by the removal of a carboxyl group at the center, leading to the formation of alpha ketoglutarate. Now then in the next step, that is oxidation of alpha ketoglutarate to succinyl coenzyme A with the release of carbon dioxide. So basically this step of tricarboxylic acid cycle involves the conversion of alpha ketoglutarate to succinyl coenzyme A, and basically this process is catalyzed by alpha ketoglutarate dehydrogenase complex. This complex, similar to the pyruvate dehydrogenase complex, consists of 3 enzymes: alpha ketoglutarate dehydrogenase, you must note it, then dihydroxylipoyl trans-succinylase and dihydroxylipoyl dehydrogenase. So this reaction proceeds in basically 5 steps requiring the 5 different coenzymes, namely thiamine pyrophosphate TPP, lipoic acid, coenzyme A, which contains a thiol group, flavin adenine dinucleotide, and nicotine amide adenine dinucleotide. So, the mechanism basically is very much analogous to the reaction catalyzed by the pyruvate dehydrogenase complex. Basically, even of alpha ketoglutarate dehydrogenase complex binds with alpha ketoglutarate and differs slightly from even of the pyruvate dehydrogenase complex in some amino acids. However, the dihydroxylipoyl dehydrogenase in both complex is more or less similar, suggesting a sort of common ancestral origin. This is very interesting point to note at this juncture. Unlike the pyruvate dehydrogenase complex, even of the alpha ketoglutarate dehydrogenase complex is not regulated by phosphorylation. Then how it is regulated? Basically, AMP serves as the activator for the enzyme here in this case. During the reaction, what happens that NADH is basically produced similar to the pyruvate dehydrogenase reaction along with the formation of succinyl coenzyme A and the release of one carbon in the form of carbon dioxide. Basically, both succinyl coenzyme A and NADH are basically rich in energy compounds and play very crucial roles as a source of metabolic energy. NADH basically can generate ATP through the electron transport chain, whereas succinyl coenzyme A is further metabolized in the TCA cycle. Now, in the next step, succinyl coenzyme A is converted into succinate. Basically, just I mentioned that succinyl coenzyme A serves as a high energy intermediate with a negative free energy of hydrolysis. This energy is utilized for substrate level phosphorylation, driving the phosphorylation of ADP to ATP in plants and bacteria and also in animals. It leads to the phosphorylation of GTP, resulting in the synthesis of GTP. The enzymatic reaction is basically catalyzed by succinate thiokinase, also known as succinyl coenzyme A synthetase. So, basically this reaction is very unique in the tricarboxylic acid cycle, as it involves substrate level phosphorylation, where energy for phosphorylation is basically provided by the substrate rather than relying on the electron transport chain or a proton gradient. We have discussed this fact while discussing the substrate level phosphorylation, because certain intermediate compounds generally have high phosphoryl group transport potential than ATP. That is why they donate phosphate group to ADP to form ATP. So, these intermediates basically have high phosphoryl group transport potential. So, the free energy of hydrolysis for succinyl coenzyme A and GTP or ATP are very much comparable, and notably this reaction along with certain reactions in glycolysis plays a crucial role in cellular metabolism. Now, oxidation of succinate to fumarate. Succinate is basically converted into fumarate by the enzyme succinate dehydrogenase, which is responsible for oxidation of succinate to fumarate. This enzyme is basically a flavoprotein enzyme linked enzyme and located in the inner mitochondrial membrane and generally functions as a part of electron transport chain. Unlike TCA cycle enzymes, succinate dehydrogenase is generally bound to membrane with its succinate binding site facing the mitochondrial matrix. This enzyme is also known as succinate ubiquinone oxidoreductase and plays a very crucial role in transferring electron from succinate to ubiquinone within the electron transport chain, resulting in the formation of trans unsaturated fumarate. In addition to this, succinate dehydrogenase is basically a dimeric protein composed of subunits with molecular mass ranging from 70 to 70 kilodalton. Basically the larger subunit is covalently linked to the cofactor within the FAD, and the enzyme contains non-heme iron. And the activity of basically succinate dehydrogenase is competitively inhibited by malonate under in vitro condition and is strongly inhibited by oxaloacetate also under in vivo condition. So, this is very important and interesting fact to note at this juncture. However, this enzyme, that is succinate dehydrogenase, is can be activated by ubiquinol, namely UBQ2, adenosine triphosphate, and ADP. The effect of ATP or ADP may be, you see, indirect, in this case is indirect, possibly involving the release of oxaloacetate. Basically, the activity of the enzyme, this enzyme in plant, that is succinate dehydrogenase, may be regulated by the energy status of the mitochondrial membrane, although you see that the physiological relevance of this regulation remain basically not clear, and how the reciprocal changes in ATP and ADP concentration of the cell plays a very crucial role, but it is yet to be revealed. Now, hydration of fumarate to malate, how this hydration takes place inside the mitochondria. Basically, you see that this hydration involves basically transhydration of fumarate takes place, and this process basically entails the addition of hydrogen and OH groups to the double bond of the fumarate, ultimately leading to formation of or you can say synthesis of L-malate. The enzyme responsible for catalyzing this reversible reaction is basically fumarase, you see in the slide, that is mentioned with dark color, and this reaction is basically very much stereospecific, with L-alanine, L-malate being the substrate for the enzyme in the reversible reaction, excluding D-malate. Furthermore, you see that aconitase performs a similar reaction in the T cycle, whereas trans addition of hydrogen and OH groups occurs across the double bond of cis aconitase. Both fumarase and aconitase play crucial roles in these stereospecific reactions within the TCA cycle. The reversible reaction catalyzed by fumarase basically indicates its thermodynamic characteristics. Now, oxidation of malate to oxaloacetate, basically this reaction is catalyzed by you see that L-malate dehydrogenase, and basically the oxidation of malate to oxaloacetate require basically energy, and basically this requires delta G of 29.7 kilojoule per mole, and then if energy is available, this synthesis of oxaloacetate takes place, and then the removal of oxaloacetate in the subsequent reaction and the oxidation of NADH and NAD drive the reaction towards the right direction. Malate dehydrogenase is the enzyme responsible for this reaction and is generally ubiquitous and with different isoforms found in mitochondria, cytosol, glyoxysomes, and peroxisomes. So, this is also very important to note at this point. You see, just recall in the chloroplast of C4 plants, particular NADP specific MDH, malate dehydrogenase is present, playing a very crucial role in the production of malate from oxaloacetate during the C4 pathway of carbon dioxide assimilation. Just a quick recall of the process. So you just recall that NADMADH basically is the isoform of is a sort of isoform of this enzyme participating in the tricarboxylic acid cycle and basically involved in the exchange of reducing equivalents between the cell compartments across the membrane and serves as a NADH shuttle. Now basically this is the diagram and showing a comprehensive overview of the 8 stages of the TCA cycle. With each step we have gone through in detail. So just a quick overview of the whole process in one go. This slide is very much important. Now what is the stoichiometry of TCA cycle? That you see that the total how acetyl coenzyme A is converted into to release NADH, FADH2 and ATP. So this slide is very important in this regard. Now generally in the process of glucose oxidation, you see that all the 6 carbons are released as carbon dioxide, and then substrate level phosphorylation contributes to the production of 4 molecules of ATP with 2 ATP generated during glycolysis and one additional an additional 2 ATP during the tricarboxylic acid cycle for 2 acetyl coenzyme A molecules. So in a total of 12 reduced coenzymes are basically produced, including 2 NADH in the glycolysis, 2 NADH in the link reaction and 6 NADH 2 FADH2 into 2 TCA cycles. So during oxidative phosphorylation, the oxidation of NADH generates a proton motive force, allowing the synthesis of basically 2.5 ATP per NADH and 1.5 ATP per FADH2. Consequently, the complete oxidation of a glucose molecule results in the production of 30 to 32 ATP molecules. So this is very important fact I would like to share with you. Now, amphibolic role of tricarboxylic acid cycle. This is also very crucial, that you see that several intermediates of the TCA cycle participates in both catabolic and anabolic reactions, which shows its amphibolic nature. Therefore, you see that we could cite some examples like 2-oxoglutarate of which is one of the intermediate of the TCA cycle, is crucial for glutamate biosynthesis and is obtained from this cycle in mitochondria, cytosolic NADPH and present in the peroxisomes. 2-oxoglutarate and is basically transported into chloroplast for glutamate biosynthesis by GS Gogate enzymes. Then another intermediate, citrate. Citrate in the TCA cycle is transported out of the mitochondria, converted to isocitrate in the cytosol and serves as a source of cytosolic 2-oxoglutarate. Then another intermediate, succinyl coenzyme A of TCA, serves as a source of carbon for porphyrin containing molecules. Then another intermediate, glutamate, serves as a precursor for various nitrogenous compounds including the amino acids. Then succinyl coenzyme A and oxaloacetate serve as a precursor for various amino acids and other compounds. Then citrate exported from the mitochondria in animals is used for the synthesis of acetyl coenzyme A by the cytosolic ATP citrate lyase, which is then used for fatty acid biosynthesis. So this is very interesting to share the amphibolic role of TCA cycle. Now this is generally a basically a diagrammatic representation of amphibolic nature of TCA cycle, which just we have discussed. You can see where the different intermediates are used and they are required as a precursor. Now, in plant cell, basically the ways of one of the ways of replenishing the intermediates is by the cytosolic auxiliary state. Basically, the plant mitochondria are characterized by specific auxiliary state transporters located in the inner membrane. The carboxylation of you see one enzyme phosphoenol pyruvate occurs in the cytosol and is then catalyzed by phosphoenol pyruvate carboxylate, generating auxiliary state. This cytosolic auxiliary state can then be transported into mitochondria to participate in the TCA cycle and maintain the flow of intermediates for anabolic pathways. This is also very crucial at this point. Now, this slide shows the basically diagrammatic representation of anaplerotic reaction, in which you just clearly observe that how these intermediates of the TCA cycle serve as a precursors of some of the biomolecules. The intermediates of TCA cycle are basically replenished by anaplerotic reactions like different enzymes they are involved. Now, how TCA cycles is regulated? This is also very crucial, like we have discussed for glycolysis. Basically, ATP and ADP functions as allosteric modulator of TCA cycle enzymes. So, basically the status of ATP and ADP is very important. So, basically enzymes involved in the TCA cycle are triggered when the ATP-ADP ratio goes below a certain level. Plant exposed to light have high ATP-ADP ratio because you see that ATP is produced during light reaction in the chloroplast. So, increased level of ATP blocks several enzymes of the TCA cycle, including citrate synthase. Reduced demand for ATP in plants during environmental stress causes basically higher ATP levels, limiting the action of TCA enzymes and citrate synthase. So, the regulation of basically these key enzymes by ATP and ADP helps to maintain cellular energy homeostasis. When energy levels are low or high ADP-ATP ratio, that basically the TCA cycle is stimulated to generate more ATP through oxidative phosphorylation in electron transport chain. Opposite to it, when energy levels are high or you can say low ADP-ATP ratio, the TCA cycle is down-regulated to prevent unnecessary energy expenditure. So, this regulatory mechanism basically ensures the efficient utilization of resources in response to energy needs of the cell. Now, there is one enzyme isocitrate dehydrogenase whose activity is how inhibited. So, isocitrate dehydrogenase is basically an enzyme involved in TCA cycle that catalyzes the conversion of isocitrate to alpha ketoglutarate. So, there are two main isoforms of isocitrate dehydrogenase, namely NADP-IDH and NAD-IDH. So, NAD-IDH is typically found in the mitochondria and its activity is dependent upon the presence of NAD. Then activity of NAD and NAD-IDH is negatively regulated by levels of ATP and NADH. Basically, these high energy molecules indicate that the cell has sufficient energy and the TCA cycle can slow down to prevent unnecessary consumption of substrate. Then mylate dehydrogenase is another enzyme in the TCA cycle that catalyzes the conversion of mylate to auxiliary state. Then MDH like IDH uses NAD as cofactor in the reaction. Similarly, NAD-IDH generally malonate dehydrogenase is very sensitive to levels of NAD and NADH. An increase in NADH levels can inhibit MDH activity. Both IDH and MDH basically are regulated by the levels of NAD and NADH in the cell. High levels of ATP and NADH basically signal a well-fueled cell, leading to negative regulation of these enzymes to slow down the TCA cycle. This regulation basically helps to maintain cellular energy balance and prevent unnecessary consumption of substrate when energy needs are already met. Now, regulation of another enzyme pyruvate into the TCA cycle. This enzyme is very crucial, and basically pyruvate derived from the glycolysis is, you see that, further oxidized to produce energy in the form of adenosine triphosphate, and the regulation of this particular process is a complex and involves several key enzymes and regulatory factors. So, pyruvate dehydrogenase complex catalyze the conversion of pyruvate to acetyl coenzyme A. This is the entry point of pyruvate into TCA cycle, and it's basically this pyruvate dehydrogenase complex activity is regulated through covalent modification by phosphorylation and dephosphorylation. Phosphorylation of pyruvate dehydrogenase complex can be inhibited by phosphorylation, which is catalyzed by a specific kinase. The kinase is basically activated by high levels of ATP, acetyl coenzyme A and NADH, indicating that there is sufficient energy in the cell. So, basically this phosphorylation inhibits the activity of pyruvate dehydrogenase complex, reducing the entry of pyruvate into the TCA cycle. Then dephosphorylation catalyzed by a specific phosphatase activates the activity of pyruvate dehydrogenase complex, and basically this process is stimulated by factors such as increased level of pyruvate, ADP and calcium ions. When basically energy levels are low, indicating a need for ATP production, then pyruvate dehydrogenase complex is dephosphorylated, allowing pyruvate to enter into the TCA cycle and then generate energy. Then this also depends upon the availability of substrate. So, the availability of substrate, particularly pyruvate itself, influences the entry of pyruvate into the TCA cycle. Then high level of pyruvate resulting from the glycolysis or other metabolic pathway promote the conversion of pyruvate to acetyl coenzyme A. Then NAD and NADH ratio is also a very crucial indicator. Basically the TCA cycle is dependent on the availability of NAD as cofactor. The ratio of NAD and NADH is an important factor influence the entry of pyruvate into the TCA cycle. High level of NADH indicate that the cell has sufficient energy, can inhibit the certain enzymes in the TCA cycle, thereby influence the flow of pyruvate into the cycle. Now, there are two more enzymes. Thiol redox regulation is also very crucial, and the control cellular processes through reversible oxidation and reduction of thiol group of the proteins. These thiol groups are basically commonly found in cysteine residues of the protein and play a crucial role in redox signaling and regulation of various cellular pathways, including the TCA cycle. So, thiol redox regulation can modulate the activity of the enzymes involved in the TCA cycle, influencing its overall function. So, how thiol redox regulation impact the TCA cycle? Question arises. Now, you see that alpha ketoglutarate dehydrogenase catalyzes the conversion of alpha ketoglutarate to succinylcholine coenzyme A, generating NADH in the process. So, thiol redox regulation can modulate the activity of alpha ketoglutarate dehydrogenase. Under oxidative condition, the thiol group of cysteine residues in this enzyme may be oxidized, leading to decreased enzyme activity. Opposite to it, under reducing conditions, this enzyme is activated, promoting the activity of TCA cycle. Then another enzyme, isocitrate dehydrogenase, which is responsible for conversion of isocitrate to alpha ketoglutarate, producing NADH. Then thiol redox regulation could influence the activity of this very important enzyme, isocitrate dehydrogenase, affecting the flow of metabolites to the TCA cycle. Then succinate dehydrogenase, also known as complex second of the electron transport chain, participate in both TCA cycle and electron transport chain. It basically oxidizes the succinate to fumarate and transfer electron to the respiratory chain. Thiol redox regulation therefore impact the activity of SDH, influencing the oral efficiency of TCA cycle and electron transport chain. Then thyrodoxin system in plant mitochondria is also very crucial. That thyrodoxin system is redox regulatory system found in various organisms including plants. Basically this involves thyrodoxin proteins, which are small proteins containing thiol groups. These thiol groups can undergo reversible oxidation reduction reactions, allowing them to act as a redox regulator. Then what is the role of thyrodoxin in TCA cycle? That you see that in Arabidopsis, a study has been conducted that how this thyrodoxin regulate the TCA cycle, enzymes of the TCA cycle. Specifically, succinic acid dehydrogenase and fumarase basically involved in the TCA cycle are regulated. When reduced by thyrodoxin, these enzymes are reduced by thyrodoxin, succinic acid dehydrogenase and fumarase are deactivated. Reduction in this context basically refers to the addition of electrons or removal of oxygen, and it often leads to change in the enzyme's conformation or its activity. Now, what are the implications of these regulation? So basically, deactivation of succinic acid dehydrogenase and fumarase suggests that the thyrodoxin system might modulate the TCA cycle by influencing the activity of these enzymes. This regulation could impact the flow of metabolites through TCA cycle, affecting energy production, and various metabolic pathways are connected to it. So it could be summarized that TCA cycle is also regulated in a very coordinated manner. So this is all about for today's session. We have gone into details of TCA cycle. Thank you very much.